inside every single cell of your body, lies one of nature's greatest engineering marvels: DNA. If we stretched out the DNA from just one human cell, it would measure nearly 2 meters in length. Yet somehow, all of that genetic information fits inside a nucleus that's only a few micrometers. You may already know that DNA forms a beautiful double helix. But here's something even more fascinating. The double helix isn't the final level of DNA organization. It minus DNA organization. DNA twists even further. This is known as DNA supercoiling. But why would a molecule need another level of twisting? The answer reveals one of the most important secrets behind DNA packaging.
Nearly 2 meters of DNA fit inside a nucleus just a few micrometers wide. But simply squeezing it in isn't enough.
Imagine stuffing a long rope into a small box. Pushed in randomly, it quickly becomes an impossible, tangled mess.
The cell faces this exact challenge. DNA isn't just stored, it folds into highly organized, accessible structures.
It must remain efficiently packed, yet instantly accessible for the cell to read genes and copy genetic information on demand. This remarkable balance is possible because DNA has an additional level of organization, far beyond the familiar double helix. Which brings us to the central question. How does DNA supercoiling actually work? To answer what DNA supercoiling exactly is, look at an old telephone cord. It is already coiled, but twisting it further makes it wrap around itself. In other words, a coil starts coiling again. DNA behaves in a remarkably similar way. The familiar double helix is already a coil. But under certain conditions, this entire central helical axis can twist and coil upon itself, creating a new level of organization called DNA supercoiling. Scientists describe this as the coiling of a coil. The original helix remains intact, but folds into a larger three-dimensional structure. Without this extra coiling, DNA is in a relaxed state. However, in living cells, most DNA exists in a supercoiled form to function properly. But what forces DNA to bend? The answer lies in structural strain, a hidden force we will explore. So, what actually causes DNA to form supercoils? It all begins with underwinding, 10 base pairs. This is the molecule's most stable configuration. Now imagine removing one of those helical turns without changing the length of the DNA. The same number of base pairs now has to fit into fewer turns, forcing the DNA away from its preferred structure. But under certain conditions, this entire central helical axis can twist and coil upon itself, creating a new level of organization called DNA supercoiling. Scientists describe this as the coiling of a coil. The original double helix remains intact, but folds into a larger three-dimensional structure. Without this extra coiling, DNA is in a relaxed state. However, in living cells, most DNA exists in a supercoiled form to function properly. But what forces DNA to bend into these supercoils? The answer lies in structural strain, a hidden force we will explore in the next scene.
To understand DNA supercoiling, scientists often study small like linear DNA. A plasmid has no free ends. Its 2 strands form a completely closed circle.
This closed structure is extremely important because the DNA strands cannot freely rotate around each other. Even if the molecule bends, twists, or changes shape, its overall topological arrangement remains unchanged as long as both strands stay intact.
But what happens if one strand is broken? Even a tiny nick allows the DNA to rotate freely, causing the stored torsional strain to disappear and the molecule to relax almost instantly.
This difference between a closed circular DNA and a nicked DNA forms the foundation of DNA topology, a branch of mathematics used to describe properties that remain unchanged. With this idea in mind, we're now ready to explore one of the most important concepts in DNA topology, the linking number, the mathematical measure that defines DNA supercoiling. Now that we understand closed circular DNA, we can introduce one of the most important concepts in topology, the linking number, or LK. A helpful way to visualize this is the soap film analogy. Imagine one DNA strand forming the boundary of a transparent surface. The second strand passes through that surface is the linking number. However, if even one strand is broken, the DNA can rotate freely, and the linking number is no longer defined. As long as both strands remain unbroken, this number never changes, even if the DNA bends or twists into different shapes.
When DNA loses turns, it becomes underwound, producing double helix, creative positive supercoiling. They have very different a structure and function.
If the DNA gains or loses helical turns, the difference is called delta LK, calculated simply as the current linking number minus the relaxed linking number. Because DNA molecules can have different lengths, scientists use another quantity called superhelical density, represented by the Greek letter sigma, or psi.
It is calculated by dividing dat lelk, allowing supercoiling to be compared across DNA molecules of different sizes. But what does a negative or positive value of sigma actually mean?
When DNA loses helical turns, it becomes underwound, producing negative supercoiling. This makes DNA easier to open. In contrast, adding extra helical turns overwinds the DNA, creating positive supercoiling. These two forms are mirror images of each other, but they have very different effects on DNA structure and function.
Changes in DNA supercoiling can be understood using two important components, twist and writhe. Twist, or twu, describes how tightly the two DNA strands wind around.
Together, these two components determine the linking number. This is expressed by the equation LK equals 2W plus WR. In other words, when DNA experiences strain, it can redistribute that strain between twist and writhe while maintaining its overall topology.
DNA supercoiling is reading cell. Understanding it brings us one step closer to understanding the remarkable machinery inside every living cell.